Hi everyone, welcome again to the course of structural biology. We are going through structural biology techniques. We have already covered the high resolution techniques like X-ray crystallography, NMR. Now we are going through different low resolution techniques in spectroscopy. We already discussed about the principle of Raman spectroscopy and how it is correlated with infrared. They both are operating in the vibrational mode. So, we have discussed their mutual exclusiveness and their differences, their similarities, their advantage, disadvantages. Today, we will discuss about how Raman spectroscopy is used their fingerprints to get information about protein structure, and more importantly, we will discuss about a very recent technique which is Raman microscopy and also a modified version of Raman microscopy which is called Raman spectroscopy. Very few people have opportunity to work using this technique. I personally have worked with that, and Raman crystallography is a technique which is amazing, which is very novel, because by using this technology you could trap a live reaction. I repeat, you could trap a live enzymatic reaction. So, let us go to and start Raman spectroscopy, Raman microscopy, Raman crystallography for studying protein. We will start with resonance Raman. Resonance Raman spectroscopy is a variant of normal Raman spectroscopy. You probably remember that in the last class I talked about Raman signal is very weak. So, normal Raman was not very fruitful in experimental purposes, in data collection. It needs very specific instrumentation. Normal Raman spectroscopy uses lesser excitation at any wavelength in order to measure the Raman scattering of this laser light. In resonance Raman, the excitation wavelength is carefully chosen to overlap with or be very close to an electronic transition. This typically means in an area of UV visible absorption. The frequency coincidence or resonance can lead to greatly enhanced intensity of Raman scattering, which facilitate the study of compounds present at low concentration. Such overlap can result in scattering intensities which are increased by factor 10 to the power 2 to 10 to the power 6. This result in significant decrease in detection limit and increase the signal intensity and decrease also the measurement time. Another technique which help Raman spectroscopy to grow is surface enhanced Raman spectroscopy or SERS, surface sensitive technique that enables Raman scattering by molecules adsorbed on rough metal surfaces. It could also be adsorbed by nanostructures such as plasmonic magnetic silica nanotubes. The enhancement factor can be as much as 10 to the power 10 to 10 to the power 11. Detection of single molecule, that is a very critical part. Using surface enhancement, Raman spectroscopy is now enabled to identify single molecule. So, in Raman spectroscopy, if you see when the breathing mode and stretching mode in the FT Raman spectrum of neat benzene, so you could get the aromatic CH stretch at 3063 centimeter inverse and ring breathing mode at 992 centimeter inverse. So, you could calculate characteristic spectra from there. Application: Raman spectroscopy is commonly used in chemistry since vibrational information is specific to the chemical bonds and symmetry of molecules. Therefore, it provides a fingerprint by which molecule can be identified. In solid state physics, spontaneous Raman spectroscopy is used to characterize material, measure temperature and find the crystallographic orientation of the sample. Raman spectroscopy can be used to investigate the chemical composition of historical documents such as and contribute to knowledge of the social and economic condition at the time the documents are produced. So, by this way you could recover old data and know about the origin and authenticity of the document. There is a huge initiative where people try to fraud about informations, historical documents, claiming that they have found papers which are from ancient ones. So, Raman spectroscopy instrumental in detecting the fraud about them. Coming to what we are for, Raman spectroscopy for protein, very relevant for this course. Secondary structure of protein, you have spatial arrangement of bonds, the amide bonds, coupling between individual vibration of CO and amide Raman bands. There are fingerprints of amide Raman bands. In secondary structure analysis, 9 normal modes are allowed for the amide band for the protein. These are called 1 to 7 and AB, the 9 amide bonds. Each of the bands shows different frequency and shift. This is because of stretching and bending. So, amide 1 band, it is majority of the contribution coming from C double bond O stretch, mostly around 1650 centimeter inverse. Different type of secondary structures are characterized by amide 1 band, slightly different in position and shape. So, you could do the assignment of secondary structure from amide 1 band. Amide 2 band contributions are coming from NH bending and CN stretching, mostly around 1550 centimeter inverse. NH bending contributes around 60%, CN stretching contributing 40%, weak band and could not be observed in absence of resonance excitation. So, about this resonance Raman and protein, I would talk at the end of these amides. Like amide 2 band, amide 3 is also talking about CN stretch and NH bend, mostly around 1300 centimeter inverse. NH bending contributes around 30%, CN stretching contributing 40%. So, if you see, you will see that alpha helix, beta sheet. So, alpha helix mostly has 1270 to 1300 centimeter inverse, random coil 1243 to 1253, beta sheets 1229 to 1235. So, that is why it is also helping in assignment. Other important spectral features comes from disulfide bridges, the SS bond, the aromatic amino acids, disulfide. Experimental studies show that the proteins whose structure contain SS bridges, Raman bends are located in the spectral range of 500 to 550 centimeter inverse. So, if you see, you will see the bands in the characteristic region. Some of the bands of aromatic amino acid in proteins are sensitive to microenvironment. You see the aromatic residues and their mean frequencies. Important Raman modes of aromatic amino acids in protein structure could be found by looking at those frequencies, and it is possible to identify. Phenylalanine shows a very intense band at 1000 centimeter inverse. So, this is the band for phenylalanine. This band is not sensitive to conformational change of the protein, therefore, could be used as a signature. You should get it with the presence of phenylalanine. Tyrosine, in case of Raman spectra of tyrosine, a doublet is observed at 830 and 850 centimeter inverse. You see this doublet. The doublets are caused by Fermi resonance affecting the in-plane breathing of phenol rings. The intensities of these two bands depend on the hydrogen bonding condition of the phenol side chain. So, you could detect the state in which the phenolic proton is present. The component of the Fermi doublet at tryptophan 1340 and 1360, I1360 by I1340 serve as a hydrophobicity marker. The I1360 centimeter inverse band is strong in case of hydrophobic environment, and the other one in 1340 in hydrophilic environment. So, I850 by I830 signifies above 4.0 non-hydrogen bonding or no hydrogen bonding, around 2.5 strong acceptor for the hydroxyl group, around 1.5 both donor and acceptor, around 0.5 strong donor. So, it is a very good signature. Histidine shows characteristic spectra at 218 and 204 nanometer. The ultraviolet resonance Raman bands are sensitive to protonation, but the enhancement is relatively weak and the histidine bands are obscured in protein resonance Raman spectra by the other aromatic contributions. This is the spectra of a protein where you could see the effect of disulfide bond. You see around 500 as we have talked about. The amino acids are shown in sky blue. You see individual amino acids are coming here. The peptide backbone in these characteristics peaks you get. So, you by putting Raman intensity versus wave number, you could identify the different modes like presence of disulfide bond, specific amino acids like especially the aromatic ones, and the change of conformation of the peptide bond. So, covalent disulfide bond could detect, hydrogen bond could detect, hydrophobic interaction could detect, ionic interaction could detect, protein structure could be altered with change in environment. The conformational change could be often observed and followed from Raman spectrum. Raman spectroscopy allow us to study folding unfolding process with more sensitivity. These experiments are now designed and planned further. Shift in amide-I and amide-II band was observed, indicating transition of the structure from alpha helical to beta sheets and to unstructured form. Here you could see Raman spectra of rice globulin under different pH condition, how the characteristic spectra are shifting. Looking at that, we could have talk about the content of the secondary structure. I talked about those characteristic bands, the fingerprints are good, but if you see in reality, the tendency or the frequency of people to study protein using Raman spectra is really less. The reason is Raman spectra, the normal Raman is basically not good, but then resonance Raman or surface enhanced Raman, which gives good intensity for small molecule or other polymers and all. For protein, there is a problem in designing those experiments, because in presence of those setup, there is a possibility of protein to be denatured. To overcome this, today I talk about a extremely new technology which have started with the concept of Raman microscopy. So, people who have no idea about microscope, just to talk about, a microscope uses source of energy and a system of lenses to magnify images of small samples. So, there are set of system, you have objective lens and you have eyepiece lens and you have many of them. A compound microscope is a microscope which uses a lens close to the object being viewed to collect energy, which focuses a real image of the object inside the microscope. That image is then magnified by a second lens, or more importantly group of lenses, which is now used in modern microscope, that gives the viewer an enlarged virtual image of the object. The use of a compound objective and eyepiece combination allows for much higher magnification. Now, I will talk about confocal microscopy. If you look at confocal microscopy picture, you see the pinhole. Pinhole is the key to confocal. So, the key part of the confocal is the pinhole. Only the light from the focal plane passes through the pinhole to the detector. So, only the light from the focal plane that passes through the pinhole of the detector. By blocking the light from outside the focal plane, confocals have good Z axis resolution. So, if you see, you could have get the width. This microscope is great for imaging thick samples, because the hedge from the out of focus object is mostly eliminated. Confocal microscope is a powerful tool for generating high resolution images and 3D reconstruction of a specimen. In confocal microscopy, a laser light beam is focused onto a fluorescent specimen through the objective lens. The mixture of reflected and emitted light is captured by the same objective and is sent to the dichroic mirror. The reflected light is deviated by the mirror, while the emitted fluorescent light passes through a confocal aperture to reduce the out of focus light. The focused light then passes through the emission filter and proceeds to the photomultiplier. In order to generate an entire image, the single point is scanned in an XY manner as a laser focus is moved over the specimen. Also, there is more color possibilities because the images are detected by a computer rather than by eye. It is possible to detect more color differences. Now, I talk about a Raman spectrometer and a confocal microscope and a microscope also. So, you couple a Raman spectrometer to a standard microscope, allowing high magnification visualization of a sample and Raman analysis with a microscopic laser spot. Raman microscopy is easy, simply place the sample under the microscope, focus and make a measurement. Just adding a microscope to Raman spectrometer does not give a controlled sampling volume. For this, a special filter is required. Confocal Raman microscopy refers the ability to specially filter the analysis volume of the sample in the XY, the lateral, and the Z, the depth axis. Coming to Raman crystallography, as I told, it is a unique concept to trap live enzymatic reaction. The Raman spectra obtained using a Raman microscope offer a unique and incisive approach to follow interactions and reactions inside a single crystal under soak in or soak out condition. First, understand the concept. You have a crystal. The crystal offer a 3D environment, right. Now, you have a setup of confocal microscope, where you could scan the Z axis. Now, imagine you are not taking the light, you are taking the Raman spectra by putting a Raman monochromator and receiving that. So, what is happening? Now, your sample is this, in you are replacing a liquid sample to a crystal. What is the advantage? First advantage is very high concentration of protein, right. If you compare between a solution of protein and the same concentration to get a crystal, at least you get 10 to the power 6 to 10 to the power 10 time. That is the enhancement. Okay. First advantage. Second advantage. So, because you get that high, you could use very weak laser or no laser. Third advantage, you are only scanning the Z axis. So, you only look at what is happening inside the crystal. Why it is so interesting? Think about, you have a small molecule A. Now, you have characteristic spectra of this A molecule. Now, you set everything, you set a protein crystal which is affecting A. If A would be changing to A1, A2, there is change of bond, there would be change of spectra, like this spectra is big, like that. So, here you align and this is a different, you understand. So, what you do, you have a high resolution CCD camera. So, the ray is coming out, you get the thing live. The data you are getting here, you have the baseline, baseline of the intact compound, and whatever is coming through you are aligning it. So, whenever you get a change, you know that this is the time when the bond is breaking or making. So, first advantages of Raman crystallography: extremely stable because you are using weak laser or no laser, under normal condition, have a low light background, provide excellent platform for Raman difference spectroscopy. What is Raman difference? As I was talking about, the spectra of compound A. Now, A is changing. Suppose it is a amide, let us say it is changing to imide, imine. There will change in the frequencies characteristic. You have this spectra of A, you collect the video and keep the new or upcoming spectra aligning with the base spectra. Whenever there is a change, you, so how you could detect? What? By doing quantum calculation, you could make what type of change, when change, when is the reaction happening, what is the span of the reaction, and everything. So, little bit talking about Bio-Raman, which is the basis of Raman crystallography. Soon after the birth of modern Bio-Raman spectroscopy in R.C. Lord's laboratory in 1960, it was realized that Raman spectroscopy could be used to compare the conformations of proteins in solution with those in single crystals. You and co-worker compared the crystal and solution Raman spectra for proteins such as alpha lactalbumin, insulin and glucagon. So, they take it in the solution, they take it in the crystal and they compare. Single protein crystals were mounted in a cuvette or glass vial and positioned by eye at the focal point of the 4 optics, that is directly at the front of a standard Raman instrument of the time. So, now I will talk about things which you will see how the changed setup give us more control. The goal is to use of a Raman microscope to follow chemical changes within single crystal of macromolecule in a manner that is synergistic with X-ray crystallographic analysis. So, basically what we are doing, we are using Raman spectra, we are using confocal microscope. So, we are using Raman spectra, especially the difference in Raman spectra. We are using confocal, so that we lock in Z of the crystal. We are using protein crystal as a platform of the experimental setup. So, Raman spectroscopy, confocal and crystallography combined, we say, we name this unique technique as Raman crystallography. A crystal is usually placed in a hanging drop which then act as the laboratory, because this is the platform for carrying out soaking in or soaking out experiment. What that means? So, you have the crystal, okay, in the drop, you add the small molecule, it goes inside and it would be only counted, it would be only visible if it comes inside, because in the instrumental setup we are only scanning the Z axis, we are not accepting any other rays, any other information which are coming from other sources. We are only locked at the Z axis using the key concept of pinhole, which is the key concept of confocal. So, see, this is the crystal we put here. Now, gradually the small molecule would be going inside the protein crystal. Now, the enzyme will react, that would change the molecule as I was talking. Whenever there is a change, there would be a change in the spectra. That change would be identified by the difference Raman, which is continuously going through by the detection of the CCD camera. So, live. So, whenever you see change, you know that this is the moment there is going a change. This is the setup. You could make changes, but this is the basic setup. So, this is the crystal tray. You put your crystal here and you have the video CCD, you have the Raman monochromator, you have laser light, you could use laser like in less intensity or do not use it. Also the crystal you have, you could see the position of the crystal and you could control it. Okay. Just like you see the microscope objective, you have the crystal here, the scattered light is coming, the laser light, here is the cover slip and this is the protein crystal. A protein is purified and crystals are grown. So, you could do that only if you have the protein crystallography setup, only if you do the every top things we have discussed in time of protein crystallography. The crystal would be placed under the microscope. Each of the crystal in the drop could be viewed by the microscope objective. To make it better, you could have put only single crystal, that makes experimental control even more. The crystal could also be viewed by using the video CCD camera, as you see it is shown here. This setup gives a certain degree of experimental control. Standard illuminating source is blocked, as I told earlier. Back scattered light from the focal spot goes back through the microscope via optical filters and a second fiber optic fed into the Raman spectrograph. So, the Raman spectrograph is continuously generated. The Raman spectral image at the CCD photon detected is then appears on the computer monitor, providing the Raman spectrum associated with the focal point of the crystal. So, advantage, very high signal to noise, the macromolecule is at its highest known concentration, typically tens of millimolar, non-resonance Raman conditions are adequate, good enough. Low extraneous light background, the contribution from luminescence, intrinsic or extrinsic from impurities are minimal in the crystal. Very stable spectral profile, they do not drift, which greatly facilitate accurate spectral subtractions in the different Raman spectra. Fine control over the desired experiment inside the crystal by varying the soak in and soak out condition. So, you could change the concentration of the small molecule, you could change the small molecule, you now have another platform to do a beautiful range of experiments. Application, as I told, the application is you could see the live reaction, but going into more detail, I have taken an example of enzyme beta lactamase where I used to work, but also the person who worked on Raman crystallography most, William C. Perry. He had worked extensively taking this enzyme beta lactamase, because beta lactamase is one of the rare enzyme which works, when I say work, I mean react with the substrate covalently. If you have gone through my protein studies, if you have started understanding enzymology, you know that enzyme just take part in the reaction, make the reaction faster, make the reaction possible, but do not take part directly in the reaction. Exception is this enzyme beta lactamase or penicillin binding proteins, they directly take part in the reaction, reacting with the beta lactams. So, that is why for this technique, for tracking a live reaction, this is the best possible vehicle. So, looking at important residues, the primary catalytic residue is serine 70, the secondary catalytic residues is lysine 73, glue 166, serine 130, lysine 234. Other important residues are asparagine 136, arginine 164 and aspartate 179. If you are new to the field, you might think that what I am talking about, how I am talking about a number, because if you get same family of enzyme, you find that they have different numbers of the residues. Yes, I agree, but beta lactamase, if you are unaware about the field of antimicrobial resistance where we work, in the field of antimicrobial resistance, beta lactamase or PBB binding proteins are the major target. More than 80% of the antibiotics are beta lactam or beta lactamase inhibitors. What this mean? This means that because this enzyme is so important, people have developed a language to talk about this, which is called Ambler's classification. What is Ambler classification? When you are talking about class A beta lactamase, you take the enzyme TAME from E. coli, record its residue numbers and talking about others, you will refer to this number. Definitely for other enzymes like in Mycobacterium tuberculosis blasi, suppose this serine is 84, but we do not call it 84, we call it serine 70 in that way. So, these are the important residues, and more interestingly, a group of drug called mechanism based inhibitors or MBIs, they are tazobactam, sulbactam and clavulanic acid. If you look at them, they have mostly similar structure. So, these MBIs take very important role in beta lactam based therapeutics, because we all know now the classic beta lactams are ineffective, there is resistance against them, but when you use them with this, it is called combinatorial therapeutics. Combinatorial therapeutics is still working for them, and especially for resistant pathogens we use them. So, here is the mechanism, and looking at the mechanism, what you want to look at, I am showing you the serine 70 is involved, but we know that serine 70 is catalytic residue. So, involvement of serine 70 is understandable, but more interestingly this MBIs, they involve a second serine, which is serine 130. That is the beauty of how these mechanism based inhibitors work. they not only involve the catalytic serine 70, they also involve a secondary catalytic residue serine 130, make a bridge between them. If you see, they make a bridge between them. So, it is very difficult to develop resistance. So, we want to, so these are good drug, they worked, but we want to know more about them, so that we could develop better drugs, next generation drugs. So, what experimental setup is developed? The reaction between three clinically relevant inhibitors, tazobactam, sulbactam and clavulanic acid and beta-lactamase, have been followed in single crystal using Raman microscope, or you could say Raman crystallography apparatus. A 647 nanometer krypton laser beam was focused onto the protein crystal suspended on the underside of a siliconized quartz cover slip. So, that is also the presence of siliconized quartz cover slip actually reduce the intensity. Laser power 120 megawatt was focused using 20x objective to a 20 micrometer spot on the crystal. The crystals and the laser spot were visualized with real time color video display to ensure alignment and that no photo damage was occurring in the crystal. During data collection, spectra were acquired over 10-second interval and 10 spectra average for each acquisition time point. So, it is giving accuracy. Spectra of the apo-beta-lactamase protein crystals were obtained, where there was no small molecule, followed by the addition of the inhibitors to the drop to achieve a final drop volume of 4 microliter and a final inhibitor concentration of 5 millimolar. Spectra were then acquired serially every 2 to 3 minutes following addition of the inhibitor. For experiment in D2O, Hippies buffer were prepared using non-deuterated Hippies and titrated with NaOD to pH 6.6. An apo-beta-lactamase spectrum was subtracted from the inhibited protein spectra at varying time interval following addition of inhibitor. According to this equation, the difference spectrum equal to protein plus inhibitor minus F protein, where F is a subtraction factor selected to minimize the protein amide one band from the apoprotein in the different spectra, because you are expecting to study the beta lactams who also have, if you look at the structure, you see they also have a CON. So, the assignments: Raman difference spectra reveals an intermediate enamine band at 1605 centimeter inverse in water, which shifts to 1587 centimeter inverse in D2O due to NH exchange with this buffer. The corresponding features of sulbactam and tazobactam are seen at 1599 centimeter inverse. And if you see now, we have used a mutant. Why we use mutant? Because we want to stop the reaction. So, there is, if you see the mechanism, there is a acetylation step and deacetylation step. We stop the deacetylation by making this glutamate to alanine mutation, and we see that when we have recorded, there is change in the span as well as change in the peak. So, before that, using kinetic studies, we were not, I would say, specific to demonstrate the effect of this 3 mechanism based inhibitor, but now we have specific determinators. So, in summary, recent studies using a Raman microscope have shown that single protein crystals provide an ideal platform to undertake Raman difference spectroscopic analysis under non-resonance conditions. This approach terms Raman crystallography provide a means of characterizing chemical events within the crystal, such as ligand binding and enzymatic reaction. In many cases, Raman crystallography goes hand in hand with X-ray crystallographic studies, because the Raman results can inform the X-ray crystallographer about the status of chemical events in the crystal prior to flash freezing and X-ray analysis. So, what it is talking about, sometime we do soaking and all, and we have no idea what actually had gone inside. So, we do the entire procedure and then we find that the substrate did not go inside, or the substrate did not, like we did not get it properly. But now if you combine them, if you do a Raman crystallography analysis before going for the final data collection, you could actually know that your drug is there or not, because you see the characteristic spectra of your small molecule in the Raman monochromata. In turn, the combined data from the Raman and X-ray analysis are highly synergistic and offer novel perspective on structure and dynamics in enzyme active site. In a related area, protein misfolding, Raman microscopy, which I did not explain much, can provide detailed insights into the chemistry of the amyloid plaques associated with Alzheimer's disease and into the intermediates of the alpha synuclein protein misfolding pathway implicated in Parkinson's disease. So, you see the normal amide bands and then you see the alteration. Looking at the alteration, you could actually comment on, and very interestingly, this could have give you the opportunity to develop live experiments on a animal to see the plaques are developing or not. So, we are at the end of this module. We have discussed the general principle of spectroscopy. We have talked about circular dichroism, we have talked about UV visible spectroscopy, we talked about fluorescence spectroscopy, we talked about green fluorescence protein, which is an amazing tool in modern age to trap live experiments, to do many tagging experiments. GF is really a gift to human civilization and our advancement in identifying a lot of biological phenomena. Then we talk about infrared spectroscopy. I agree that infrared is not much to do with protein, but infrared and Raman, as we have talked, they are getting fingerprints, they are getting significant interesting data, which in future with modern setup, moderated setup where we could study proteins would give us more information. And we finish up with Raman. We talk about Raman's effect on its characteristic spectra of protein, but more importantly Raman microscopy and Raman spectroscopy, the two Raman crystallography, the two techniques which people are looking for in coming days. With that, I would finish this module. Thank you very much for listening.